Hello everyone, this is our BIS103 map part 4, which will be accompanying our lectures 11 and 12 and get us ready for those lectures where we will be discussing the ETC or the electron transfer chain. So what I want to do in this map is to really summarize the lectures 1 to 10 and all the pathways that we discussed so far, leading up now to our ETC, where we finally want to make ATP. So what have we accomplished so far? We started out with our core glycolytic pathway here, going from glucose down to pyruvate. We also had discussed how we can bring other monosaccharides and disaccharides, other carbohydrates into the glycolytic pathway for their breakdown for generating chemical energy. We also had discussed our polysaccharides, glycogen and starch, and how these can be broken down and fed into the glycolytic pathway, depending on whether they're internal storage or whether we are getting them through our diet. And then we had discussed how all of this pyruvate that is coming from the breakdown of these carbohydrates can be converted in our PDH reaction here into acetyl-CoA. And then acetyl-CoA, we had discussed how it can enter the TCA cycle and can then either be decarboxylated in the TCA cycle to generate our reduced electron carriers that we will see in the next two lectures we are needing to do our ETC, or we can use acetyl-CoA through the TCA cycle to generate a number of other metabolites that can be used as biosynthetic precursors. In addition, right, we had talked about what happens actually if we don't have oxygen. Right? All of the pathways that I had shown you right here in the map are dependent on the presence of oxygen. If we don't have oxygen, right, we had talked about two specific examples on how we can use fermentation to bring about the breakdown of pyruvate, specifically important here to regenerate our cofactors. And two specific examples were lactate and ethanol, where right, we said we cannot do ethanol, mammals cannot do it. In this case, it would be lactate fermentation. And then we started to turn things around, right, and had moved on a bit from our catabolic type pathways to more the anabolic type pathways. So for example, we discussed our TCA refill reactions, the anaplerotic reactions, on how we can refill the TCA cycle if we are removing metabolites for the formation of other biosynthetic precursors. We had discussed our GNG, or gluconeogenesis, right? How we can use non-carbohydrate metabolites such as amino acids or lactate to generate hexoses and potentially free glucose through our gluconeogenic pathway, so backwards in the glycolytic pathway. We also had discussed the Cori cycle, right? How we can use, for example, lactate that is formed in the muscle in the absence of oxygen in muscle cells, how it can be transported to the liver and there reconverted to hexoses and specifically glucose through our gluconeogenesis pathway. And then we had discussed specifically the liver has this one enzyme here, this phosphatase, that allows us to convert glucose 6-phosphate into free glucose. And gluconeogenesis typically will end in glucose 6-phosphate for all the other purposes that you want to use this for. But the liver's job was to release free glucose to the bloodstream. So the liver has a specific phosphatase to actually dephosphorate glucose 6-phosphate. So that's only happening there. Then we had also looked at how can we go further past the production of glucose in gluconeogenesis and to actually go all the way to glycogen. And we had talked about the pentose phosphate pathway, our PPP, how this intersects now at this key branching point of these pathways at glucose 6-phosphate, and how we can use the PPP, for example, for the metabolism of C4, C5, and C7 sugars, for the production of a reducing agent glutathione, and for the production especially of another reducing cofactor, our NADPH. Then we had switched gears again. We started to talk about our lipid, fat, and fatty acid metabolism. We had talked about how we can use beta oxidation to break down fatty acids into acetyl-CoA. And again here, we had also turned it around and started to talk about how we can actually use acetyl-CoA for the biosynthesis of fatty acids. We had talked about how it is important that the PPP intersects with these pathways because it's providing the critical NADPH cofactor here, for example, for fatty acid synthesis. This wasn't the only role for NADPH, but one of the major ones. And at the end, we also had discussed one exception here, the glyoxylate cycle, a cycle that animals don't have, but plants, microbes have it and can actually bring about the net conversion of acetyl-CoA from fatty acids to generate glucoses. Something we cannot do, but these organisms can do this to utilize lipid storage for the generation of sugars. So I hope this sort of pathway overview at this stage will help you as we're moving into our electron transfer chain, as we're making ADP and moving forward in our course material. So it really is meant to provide you with an overview of all the pathways that we have seen so far and how they're operating in context with each other.